CTC 210. This is your first forensics laboratory experiment. And it's not going to take very long. It's going to be quick and normally I would say painless, but maybe not in this case. Because what you're going to do is you're going to discover your own blood type. Are you A, AB, B, O, positive, negative? Which one of the 8 do we have in our classroom? Now, if you do know your blood type, great, good for you. You're going to prove that you know your blood type. So we're still going to milk your blood, and we're still going to put it in the tray, and we're still going to put the antigen on it to see if we can get it to react. That way you can see that process in person. All right, so here's how the lab is going to go down. Number 1, we have a finger prick. And we're going to make you prick your finger with the lancet. It's not going to hurt. You'll feel it, but it's not going to hurt. And we're going to have to get a couple of drops of blood from you for each test. So the best way to make sure that this happens is to take your finger and just squish it, rub it, get it warm, get it moving. We want the blood circulating as much as possible. And normally when you do that and you give it some aggravation a little bit, you can get more blood from your fingertip. There's going to be 2 different types of people in the classroom. One person, you're going to be able to prick your finger and you're going to be able to bleed freely. You will be able to get a lot of drops, and that's okay. That's what we want. The other person, you might prick your finger and you might squeeze and squeeze and squeeze and squeeze, and you can't hardly even get 1 drop out of your finger. That's kind of normal sometimes, especially if you don't warm up that finger before you decide to milk it. But that also means that you have somewhat of a thicker blood. And that also could mean that you might have high blood pressure. Or you might need to be on blood thinners in order to improve your circulation and your blood pressure numbers and your pulse and everything else that comes along with it. If you're a free bleeder, well, you could already be on blood thinners. Are you on medications? If so, that could have an effect. So what you're seeing on the screen right now are a couple of different wells, anti-A, anti-B, and anti-D, that they're going to call it, and control. The reason I'm showing you this diagram is because it shows you a very clear-cut picture of what's going to happen if your blood coagulates. That's what we're looking for. If you look in anti-A well, the anti-A well is coagulation here. Imagine you on an operating table and the doctor gives you the wrong blood. This is what's going to happen on the inside of your body. That is not what you want, right? So this means that that is a positive result for that particular blood type. Anti-A means that this person is the A blood type. You reacted with it. It reacted against your A proteins on your red blood cells. Anti-B, well, your blood type did not react with anything here. And it stayed liquidy, and it stayed watery. You didn't see the coagulation like you did here. And then for us, we have RH, but they've labeled this as anti-D. So this is going to be an A positive blood. If you were AB, the coagulation would happen in A and it would happen in B. If you were O, then you would not have coagulation in anti-B like you have right here, and you would not have coagulation in anti-A because you don't have either protein. All right? So that's what's going to happen in this lab. Number 1, you're going to prick your finger. I know it doesn't sound appealing, but it will be quick. You will hardly feel it, I promise. And you're going to milk yourself with a couple of drops into each well that we provide you. Then you're going to take the anti-serum and you're going to add a few drops of the anti-serum into each of the wells. That's step number 5. Obtain the anti-A, anti-B, and anti-RH serum and add 2 to 3 drops of each solution into the proper wells. You're going to take a clean toothpick for each one. You're going to give it a good mix, and then you're just going to kind of let it sit out for 1 or 2 minutes and watch what happens. If it's harder to see, we'll put it on our microscope and we'll take a look at it. We've got no problem with that. But we're going to look for that coagulation picture that we saw just a few seconds ago. Number 9, it's going to say take a picture of your blood well to prove that you did it. And then you're going to upload that picture into the Blackboard system. So most of you have cell phones, and this is the time where cell phone comes actually handy. So take a picture of your blood well with your cell phone and then upload it into the Blackboard system so we have proof of what you did, and we can double check your answers to make sure that you read your blood type the proper way. On the data sheet, you're probably going to see something that says like observations. Just write down what you see. Did it clump or did it stay watery and liquidy? That's all that we're after. And then here's your blood type. We want you to circle it. So what did you discover? What is your blood type? You will know by the end of the day. It will just take a couple of minutes, and that's it. The next part of the procedure is going to be some simulated blood. We're going to have some blood into the laboratory. You will not know what that blood is going to be or what it will type to be, but we will know. We've kind of, in a way, made it. And we could also give you random blood samples that we've collected from our phlebotomy class that we offer at Cape Fear. So we'll give you an assortment of different ones to test. You'll do the same thing. You'll put a few drops into each one of the wells, and then you'll add some of the anti-serums into each one of the wells, and then you'll write down your observations to see what you get. Did they come out to be A's, B's, O's, or AB's? And were they positives or were they negatives? And that's really it. That's all that we're doing in this very first lab experiment. It's pretty straightforward. It's just to get you used to this idea of blood and maybe how blood relates to you, and maybe blood typing some other samples that aren't necessarily yours, that we don't have to milk people for. Phlebotomy will do that for us. Okay? The other thing that I want to mention here is that some of these labs will have questions at the very end. You'll see this section that will say lab questions, and it says please complete them, and it's going to be graded as part of your lab exercise. So go through, answer these questions before the end of the day, and that way you can finish that and turn it in. And then you're going to see assignment and worksheet questions that will typically follow some of these labs. This is going to be somewhat like an online assignment. It will be graded as an online assignment. And they're just a couple of questions. They make great study guides. You'll go through, answer them, provide some definitions, and it just kind of sums up lecture-wise what is going on. So a part of these questions will be what's happening in the lab, and the other part of these questions will be what's happening lecture-wise to make it happen. That's what you'll see with most of the labs this semester. Maybe not every one of them, but this is the type of template that you're going to see. So for this lab, you should be able to finish the lab and answer the lab questions at least on the same day. Take a look at your calendar to see when the due dates are, and then for the assignment questions, the worksheet part, that's more lecture-driven than lab-driven, take a look at that due date as well in your calendar and turn it in before the due date happens. Okay? One thing that I want to say with the assignment is this diagram here. I've always had questions what that diagram represents. These diagrams represent coagulation. What you're seeing here are clumps. Those clumps represent coagulation. What you're seeing here, there's no clumps. These are blood cells that are not clumping together, so no coagulation is existing. That's how you need to read these diagrams, and that's what they mean. So on the last page of this, they're going to want you to read the blood wells and identify if the blood type is AB, ABO, and then positive or negative. And that's how you will determine that. Look at the clumping, and that is why they are grouped together this way. That is representation of coagulation, which, if I go back to the very beginning of the picture, that represents the cells that look like this. That is coagulation. So if you've got questions, feel free to ask. We'll help you out in the lab as much as possible. If you don't want to prick your own finger, we'll do it for you. We'll put the lancet on it. You'll never know that it happened. However, we are working with blood. That means that nothing goes in the trash can. These are biohazards. We have a special biohazard trash can that we will use. Everything that comes in contact with your blood sample needs to go into the biohazard bag. We need to be very particular with the way that we dispose of the waste that's generated from this lab. We've not really had to do that before up until now, but now we do. All right, so we'll see you in the lab. Everything will be ready by the time you get there. It won't take very long at all for the procedure, and you'll have enough time to finish out the questions and turn everything in before you leave in one sitting. Good luck.